Good morning, everyone. I am delighted to welcome you to today's presentation. My name is Dr. S.K. Singh, and I hold a PhD in this fascinating field. Today, I will be talking about the qualification of gas chromatography, mass spectrometry, or GCMS. Over the course of this presentation, we'll explore the fundamentals, applications, and significance of this powerful analytical technique. I'm excited to share insights with you, so let's get started. Today, I want to introduce you to gas chromatography, mass spectrometry, or GCMS for short. This technique is a powerful analytical tool that combines two methods, gas chromatography and mass spectrometry, to identify and quantify compounds in complex mixtures. Basically, we first separate the components using gas chromatography, which allows us to analyze each compound individually as they pass through a column. Then, as each compound exits the column, the mass spectrometer helps us identify it based on its unique mass-to-charge ratio. This synergy makes GCMS incredibly valuable in fields like environmental analysis, forensic science, and even pharmaceutical research, because it provides both separation and detailed identification in one integrated process. Throughout our presentation, you'll see how GCMS plays a crucial role in advancing our ability to analyze substances with precision and confidence. Let's take a closer look at the principle of GCMS. Essentially, it combines two powerful techniques, gas chromatography for separating complex mixtures and mass spectrometry for detecting and identifying the compounds. This combination allows us to analyze very small amounts of substances with high precision. Some of the main advantages include its high sensitivity, meaning we can detect trace levels of analytes, and its ability to provide structural identification, helping us understand the molecular makeup of what we're analyzing. It's especially useful for trace analysis, where detecting tiny quantities is critical. Now, in terms of applications, GCMS is widely used for analyzing residual solvents, as outlined in ICH Q3C guidelines, as well as pesticide residues and volatile organic compounds, or VOCs. These are typical areas where accurate, sensitive analysis is essential. The process begins with vaporizing the sample so it becomes a gas. Then, this vapor is injected into the GC column, where separation occurs based on different affinities of compounds to the column material. After separation, molecules are ionized in the mass spectrometer, either through electron ionization, EI, or chemical ionization, CI. The ions are then sorted in the mass analyzer based on their mass-to-charge ratio, or MZ. Finally, the detector captures this data, allowing us to generate spectral fingerprints for identification. This entire process enables precise, detailed analysis, making GCMS an indispensable tool in analytical chemistry. Let's take a closer look at the analytical instrument qualification lifecycle. According to the USB AIQ model, there are 4 key stages to ensure our instruments are properly qualified. First, we have DQ, which stands for Design Qualification, where we verify that the instrument's design meets our specific requirements. Next is IQ, or Installation Qualification, where we confirm that the instrument has been installed correctly and according to specifications. Then comes OQ, Operational Qualification, which involves testing to ensure the instrument operates as intended under normal conditions. Finally, we have PQ, Performance Qualification, where we evaluate whether the instrument consistently performs over time and under real-world conditions. But it's important to remember that the life cycle approach doesn't stop there. It also includes ongoing risk assessment to identify potential issues before they become problems. Periodic reviews help us stay aligned with changing standards or process needs. Preventive maintenance is key to extending the lifespan and reliability of our equipment. And requalification ensures that the instrument continues to meet all qualification criteria after any significant changes or over time. By following this life cycle approach, we create a robust framework that not only qualifies our instruments initially, but also maintains their performance and compliance throughout their entire operational life. This comprehensive strategy is essential for ensuring data integrity, product quality, and regulatory compliance in our work. Let's take a look at this example. When we're dealing with residue solvent analysis, the detection limit is a critical factor. In this case, the limit of detection, or LOD, is set at 1 ppm. This means our instrument must be capable of detecting extremely small quantities, specifically, a sensitivity of at least 10 to the negative 12 grams. That's incredibly sensitive, but essential for ensuring accurate analysis at such low levels. Now, to make sure our instrument can meet these stringent requirements, we need to focus on design qualification, or DQ. This process guarantees that the instrument is designed to fulfill the intended analytical needs. And to define those needs clearly, we often create a user requirement specification, or URS. Think of it as a detailed blueprint that outlines exactly what the instrument must do, like supporting that high sensitivity we just discussed. So, in summary, setting these detection limits and qualifying the instrument through DQ and URS ensures we get reliable, precise results for residue solvent analysis. It's all about making sure our tools are up to the task for such demanding analytical work. Now let's talk about the installation qualification, or IQ, for the GCMS. This step is crucial because it verifies that the instrument has been properly installed and is ready for reliable operation. During IQ, we check several key aspects to ensure everything is in place. First, we verify the instrument model to confirm that the correct equipment has been delivered and installed as specified. Next, we look at the electrical supply, making sure it's within the acceptable range, typically 230 volts with a plus or minus 10% tolerance, to prevent any power-related issues. We also verify the gas supply, specifically the helium purity, which should be at least 99.999%. This high purity is essential for accurate and consistent results. Additionally, we confirm that the computer and software are correctly installed, functioning properly, and ready for data acquisition. Finally, all these checks are documented thoroughly. Proper documentation not only ensures compliance, but also provides a record for future reference and troubleshooting. By completing these IQ checks, we're setting a solid foundation for the instrument's performance and overall reliability, which ties directly into our goal of maintaining high standards throughout the analytical process. Now, let's talk about operational qualification, or OQ for short. This is a crucial step in ensuring that our instruments are functioning correctly. Essentially, OQ verifies that the instrument operates within the manufacturer's specified parameters. It's like a checkup to confirm everything is working as it should. By conducting OQ, we make sure that the equipment performs reliably under real-world conditions. This step helps us identify any deviations early on, so we can address them before moving forward. Remember, maintaining this standard is vital for the overall quality and accuracy of our processes. So, in the big picture, OQ is all about confirming that our tools are up to standard, supporting the integrity of our entire operation. Let's take a closer look at OQ calculation and its role in mass accuracy. Essentially, OQ, or overall quality, helps us evaluate how accurately our measured mass matches the theoretical mass. When we're working with mass spectrometry data, precision is key, and that's where mass accuracy comes into play. By calculating OQ, we can determine how close our observed values are to the expected ones, which is vital for reliable identification. This calculation involves comparing the measured mass to the known or predicted mass of a compound, often expressed in parts per million, or ppm. The lower the ppm difference, the higher our confidence in the measurement. This is especially important in complex samples, where small deviations can lead to misidentification. By understanding and applying OQ calculations, we ensure our data's integrity and improve the reliability of our results. It ties into the overall theme of quality control in our analytical process, helping us maintain high standards and accurate interpretations throughout our work. Now, let's take a closer look at the sensitivity test calculation. This is a crucial step because it helps us understand how sensitive our model or system is to changes in input variables. Essentially, we're testing how small variations can impact our results, which is vital for assessing reliability and robustness. When performing these calculations, we typically adjust one parameter at a time to see how it influences the outcome, keeping everything else constant. This way, we can identify which factors have the most significant effect. Connecting this to our overall theme, sensitivity testing ensures that our decisions are based on stable and dependable models, giving us confidence in the results we present. Let's talk about the linearity test, which is a key part of performance qualification. This test helps us verify that our analytical method provides consistent and proportional results across a range of concentrations. Essentially, we're checking whether the response of our instrument or method is directly proportional to the amount of analyte present, from the lowest to the highest levels we're interested in. By conducting this test, we ensure that our method can accurately quantify samples throughout its working range, which is crucial for reliable decision-making. Highlighting this point connects to the overall theme of method validation, making sure our process is both reliable and robust. So, in summary, the linearity test confirms that our measurements are trustworthy across different concentrations, reinforcing the quality and accuracy of our analytical results. Let's walk through a practical example of how regression calculation works. On this slide, you'll see an example that illustrates the key steps involved. First, we start by identifying our data points and the variables we're analyzing. Next, we use these data points to compute the regression coefficients, which essentially tell us how much our dependent variable is expected to change with a unit change in the independent variable. This process involves some calculations, like finding the slope and intercept, that are fundamental to understanding the relationship between variables. Remember, the goal here is to create a model that best predicts our outcome based on the input data. By working through this example, I hope it clarifies how these calculations are applied in real-world scenarios. This is a crucial part of our overall theme, using data and statistical tools to make informed decisions. So, as we go through this example, keep in mind how these concepts connect to broader applications and the importance of accuracy in our calculations. Let's talk about the system suitability test, or SSD, which is a crucial part of our analytical process. We perform this test before starting any routine analysis to ensure that our system is working properly. It's like a quality check to make sure everything is in optimal condition. By doing this, we can confirm that the system's performance is consistent and reliable, which is vital for accurate results. Remember, the SSD helps us identify any issues early on, preventing errors in our actual sample analysis. Overall, it's an essential step to maintain the integrity of our data and uphold high standards in our analytical methods. Now, let's talk about when requalification is actually required. Essentially, after any major repair or detector replacement, it's important to perform requalification to ensure everything is still functioning correctly and safely. The same applies after a software upgrade, as updates can sometimes affect system performance or calibration. Additionally, even during routine preventive maintenance, requalification should be considered to maintain optimal operation. Remember, these steps are crucial to keep the system reliable and compliant with safety standards, which ties back to our overall goal of ensuring consistent quality and safety in our processes. Let's talk about documentation requirements, which are crucial for maintaining compliance and ensuring data integrity. Essential records include the User Requirements Specification, or URS, which outlines what the system needs to do. We also need protocols for installation qualification, IQ, operational qualification, OQ, and performance qualification, PQ, to verify that equipment and systems are functioning correctly. Raw data and reports are fundamental because they provide the evidence of actual results and activities. Calibration records are necessary to confirm that instruments are measuring accurately over time. Additionally, keeping a preventive maintenance log helps track routine upkeep, preventing issues before they occur. Overall, these documentation elements are vital for demonstrating that our processes meet regulatory standards. Remember, compliance isn't just about having the records. It's about ensuring data integrity and traceability of electronic records. That means being able to track every change or update to data to maintain transparency and accountability throughout the process. Let's take a moment to look at the lifecycle approach, which is fundamental to maintaining high-quality GCMS operations. We start with design qualification, or DQ, making sure that the instrument is designed to meet our specific needs. Next is installation qualification, IQ, confirming that everything has been installed correctly and according to specifications. Then we move on to operational qualification, OQ, which verifies that the instrument functions properly within its intended operational parameters. Following that is performance qualification, PQ, where we demonstrate that the instrument consistently produces accurate and reliable results in real-world conditions. And of course, this isn't a one-time process. Continuous monitoring is essential to catch any deviations early and ensure ongoing performance. This lifecycle approach is absolutely essential across various fields, whether we're talking about pharmaceutical quality control, environmental testing, or food safety laboratories. It helps us maintain integrity and compliance throughout the instrument's life. Now, focusing on GCMS qualification specifically, it plays a critical role in ensuring regulatory compliance, which is vital in our industry. It guarantees that our analytical results are reliable and traceable, giving confidence in the data we produce. Plus, it supports long-term instrument performance, reducing downtime and maintaining consistent quality over time. Overall, thorough qualification and ongoing monitoring are key to delivering trustworthy results and meeting the strict standards our work demands. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation useful and insightful. Remember, we're committed to supporting your success, so don't hesitate to reach out if you need anything. Thank you once again for being here and for your interest in this topic.